Your gut may be the key to beating type 2 diabetes. In this eye-opening video, we explore the latest research on gut microbiota and type 2 diabetes complications. Discover how you can improve your gut health and reduce your risk of this condition. Are you ready to take control of your health? Watch now. The crucial connection between gut microbiota and type 2 diabetes complications. The gut microbiota, a diverse community of microorganisms residing in the gastrointestinal tract, is gaining recognition for its pivotal role in human health. Comprising trillions of bacteria from over 1,000 species, this intricate ecosystem influences various physiological processes and significantly impacts our overall well-being. Dysbiosis, a state of imbalance within the gut microbiota characterized by reduced diversity and abundance of specific bacteria and fungi, has emerged as a critical factor in various health conditions. This article delves into the profound influence of gut microbiota on one of the most prevalent metabolic disorders globally, type 2 diabetes and its associated complications. Type 2 diabetes is a chronic, non-communicable disease marked by elevated blood sugar levels. It typically arises from insulin resistance, where the body's cells become less responsive to insulin's glucose-regulating effects, or insufficient insulin production by the pancreas. This metabolic disorder affects millions worldwide, with the number of cases steadily on the rise. Research has illuminated a strong connection between the composition of the gut microbiota and the development of type 2 diabetes. Dysbiosis within this microbial community can lead to insulin resistance and inflammation, key contributors to diabetes onset. Furthermore, the article uncovers specific bacterial taxa that are associated with an elevated risk of diabetes, emphasizing the importance of microbial balance. While certain bacteria may pose a higher risk of developing type 2 diabetes, others offer protection against the disease. Beneficial bacteria like Akkermansia muciniphila and specific strains of Bacteroides have been linked to improved glucose metabolism, reduced inflammation, and enhanced gut barrier function. These findings highlight the potential of harnessing the gut microbiota to mitigate diabetes risk. Dysbiosis of the gut microbiota plays a central role in the development of insulin resistance, a hallmark of type 2 diabetes. This dysregulation triggers chronic inflammation, disrupts bile acid metabolism, alters energy extraction from food, and influences the secretion of gut hormones known as incretins. These factors collectively impact insulin sensitivity, driving the progression of diabetes. Medications' impact on the gut microbiota. The article also explores how medications used to manage type 2 diabetes can influence the gut microbiota. Notably, metformin, a commonly prescribed drug, has demonstrated the ability to modify microbial composition. Metformin interacts with the gut microbiota by modulating inflammation, enhancing glucose homeostasis, improving gut barrier integrity, and promoting the presence of bacteria that produce beneficial short-chain fatty acids. Poorly controlled type 2 diabetes and associated metabolic disorders, including impaired lipid metabolism, oxidative stress, and hypertension, can result in microvascular and macrovascular complications. Microvascular complications affect small blood vessels, leading to conditions such as diabetic nephropathy, diabetic neuropathy, and diabetic retinopathy. Macrovascular complications involve larger blood vessels and include cerebrovascular disease, coronary heart disease, and peripheral vascular disease. Intriguingly, gut microbiota dysbiosis extends its influence beyond the onset of type 2 diabetes, also contributing to complications associated with the disease. Studies have detected differences in the gut microbiota composition between individuals with diabetes-related complications, such as diabetic nephropathy, retinopathy, neuropathy, and various cardiovascular conditions, and healthy controls. The severity of gut microbiota dysbiosis appears to correlate with the severity of these complications. The article delves into strategies aimed at restoring gut microbiota balance to mitigate type 2 diabetes and its complications. These approaches encompass dietary modifications that encourage the proliferation of beneficial bacteria through the consumption of fiber-rich diets, fruits, vegetables, whole grains, and fermented foods. Additionally, probiotic supplementation, prebiotics that nourish beneficial microbes, symbiotics combining pre- and probiotics, and, in extreme cases, fecal microbial transplantation holds promise for achieving microbial equilibrium. Probiotics, containing specific beneficial strains such as Lactobacillus fermentum or Bacteroides fragilis, have shown potential in enhancing glucose metabolism and mitigating inflammation in individuals with type 2 diabetes. While further research is necessary to establish their effectiveness and optimal usage, they represent a potential avenue for diabetes management. Chronic inflammation emerges as a pivotal mediator in the intricate relationship between gut microbiota and type 2 diabetes. Dysbiosis-induced inflammation contributes to insulin resistance and metabolic dysfunction, perpetuating the cycle of diabetes and its complications. Understanding this inflammatory aspect is crucial for developing targeted interventions. Finally, the article highlights the ongoing research in this field. Scientists are dedicated to unraveling the specific mechanisms by which the gut microbiota influences type 2 diabetes, developing precise microbiota-based interventions, and exploring the potential of personalized microbiome therapies for diabetes management. Additionally, researchers aim to identify novel biomarkers for assessing diabetes risk based on gut microbiota composition. In conclusion, the article underscores the indispensable role of the gut microbiota in the development of type 2 diabetes and its associated complications. Dysbiosis within the gut microbial community can exacerbate insulin resistance, inflammation, and metabolic dysfunction, contributing to the severity of diabetes. However, the potential for intervention exists through dietary modifications, probiotics, prebiotics, symbiotics, and fecal microbial transplantation, offering hope for better management and prevention of this widespread and debilitating disease.